Today we are starting with the detailed structure of stamens. In the flower of angiosperm, the stamen is the male reproductive organ. Stamen consists of three components, that is filament, anther lobe, which is fertile part, and connective. This anther lobe is connected with that of the filament with the help of connective. Filament is long and sterile structure, that is structure of stamen. Then the anther contains two lobes, that is first lobe and second lobe. This is first lobe, this is second lobe. This two-lobed structure is known as bilobed or diathecous structure. Each lobe is again divided into two sacs, first, second. These sac in each lobe is known as microsporangia. We have seen in detail, that is first sac, second sac. These sac is known as microsporangia. As a single anther is made up of 4 pollen sacs or microsporangia, also known as tetrasporangiate. Then how this microspores is developed inside the anther we discuss in detail in T.S.of anthers or transfer section of anthers. With the help of knife or blade, we cut the anther in a transverse section. That is, we have to cut this anther lobe in a transverse section. Internally, the anther consists of two types, that is anther wall and second is sporogenous tissue. We discuss the structure of anther in detail. That is, outermost protective covering of anther is known as epidermis, which is single layered structure. It is made up of tangentially stretched and flattened cell. It performs the function of protection, that is outermost layer, epidermis. Below the epidermis, endothecium or endothecial layer is present. These cells are radially arranged with fibrous bands. This layer is again single layered, known as endothecium. This layer is hygroscopic in nature and helps in the dehiscence of anther and discharge of a pollen grain at the time of dehiscence. Then below this layer, presence of Middle layer. This middle layer is parenchymatous, and this parenchymatous layer is 2 to 3 in their layers. These are temporary layers and get degenerate due to hygroscopic nature of endothecium layer. Then below that layer, presence of a circular structure, and this circular structure is again covered with a single layered surrounding structure known as tapetum. These are the innermost layer of this anther. This tapetum is present around the sporogenous tissue. This layer is physiologically important because this layer provides food material to the sporogenous tissue. This layer is multi-layered, and this layer provides nutrition to the developing pollen grain or sporogenous tissue. Then, that is, we have seen the anther wall structure. First is epidermis, which is single layered and it is protective in nature. It provides protection. Then second layer, that is endothecium. This endothecium is hygroscopic in nature and helps the dehiscence of pollen grain with a fibrous banding. That is presence of fibrous banding in endothecium layer. Then third is middle layer, which is temporary layer and get degenerated at the time of hygroscopic nature of that endothecium layer. Then below that layer, presence of the tapetum layer, and this tapetum layer provides nutrition to the developing pollen grain, and at the time of